All right, so today I'm going to be talking about using rodent models, largely mice. And I should have put in my title that this is for simple tox studies and efficacy testing for novel anti-cancer therapeutics, although our core is able to do other sorts of investigations, but we are largely set up for doing anti-cancer studies. I'm a research associate professor in CPB at Purdue. And by training, I'm a T-cell immunologist. And my current work, I investigate the immunosuppressive environment, particularly the myeloid-derived suppressor cells that shut down T-cell responses, helping to allow the cancer to grow. But I also direct this core and get into a lot of other studies that you'll see here. So I'll be sharing a lot with you about drug discovery kind of in general, just so we're all on the same page, and just some information about mice so you can be using them for yourselves in appropriate ways, or if you wanted to come to us and have us do studies for you, there's also that information in the talk. So the name of our core is the Biological Evaluation Shared Resource. We're in the Purdue Institute for Cancer Research, located on the West Lafayette campus. We're in the Hanson Life Sciences building, and that's my contact. So if you care to ask questions about these things, or if you wanted us to do any projects for you, that's how you'd get a hold of us, get a hold of me as I direct the core. Besides me, we have 3 full-time staff and 2 student workers doing the work and keeping the studies going. And our mission is to provide simple drug toxicity testing and the proof of concept efficacy studies to really help investigators, provide them a competitive edge on their grants and in publication of their manuscripts for their novel drugs or their novel therapies they've come up with. That really, to get good cancer funding now, you almost required to have animal studies in it. And as part of this mission, I also highly interface with the PIs and guide them through selecting rodent models if they need that help, and to help design the studies to make sure that we obtain the data that they want, get the answers they want, or at least get good answers that are robust and reproducible. And lots of times I'll get involved in the literature review with them to make sure we're on task. And then I also do a lot of grant preparation. Most often that's really getting involved in critiquing it and helping to set the animal portions up the way that I think is best. Or sometimes I'll even write the animal sections as well. And we have quite a good track record of getting funding. And so I really get involved in a lot of these projects quite deeply. And the people that usually come to me for help are chemists and molecular pharmacologists, biomedical engineers, lots of times the people that know they need to use animals but aren't entirely sure how to do it or don't have the bandwidth to do it. And frequently many of them will know how to use animals. That just need some extra hands in generating data. So just to briefly cover the drug discovery steps, the first starting step obviously is going to be the benchtop testing, which is going to be on cell lines like high throughput screening or microsome assays, metabolic studies, getting some idea what the toxicity might be. And then after you've found some hits and maybe developed into a lead, then you would come into needing to do animal testing. Usually that's going to start at the mouse level. Doesn't have to, but usually does. And then after mouse studies are successful or rat studies, then move on to non-rodent models, which are usually required for FDA approval for getting an IND, which would then lead you to the next step, which would be clinical trials. And then after that, the FDA reviews those clinical trials and decides an approval. And then after that, there's still, of course, the post-market safety monitoring that goes on. So we're talking about back up here in step 2, the beginning of step 2, largely using mice, but rodents particularly. And so why are mice such a good model to use? Well, their genome is remarkably the same as humans. Immunologically speaking as well, they're remarkably similar. There are several cytokines the human cytokines that will work in the mice, and of course there's many that don't. But even for the ones that don't, the species-specific cytokines are almost always doing the same things, using the same mechanisms as what's found in the humans. And however, one thing to remember, of course, is that 1% difference really can have some large consequences, as mice phenotypically don't look remotely like us. And there are certain metabolic pathways that are very different. But largely, they're a great place to start for getting initial answers. And also because they have such a short gestation period, comparatively speaking, for other mammals. Of 20 days, they're fertile at 8 weeks, and they're considered a full adult somewhere around 12 weeks. They're good breeders, and versus larger animals, they're comparatively a much lower cost. You can put 5 per cage, and those sorts of considerations make them a little less expensive. They're very easy to handle. You can hold them and restrain them with one hand. And I think perhaps most importantly is if you're going to be doing syngeneic studies, that is mouse studies with mouse cell lines, they have the most developed reagents for really deep investigations, for especially with considering the genetically engineered mouse models that are available for discovery or for investigating drug pathways or disease pathways, things like that, and as well as antibodies for detecting antigens and in-depth studies in that regard. And so just a picture of the common mouse strains that are used in drug testing. We've got C57 black 6 mouse and Balb C, and which are inbred wild type mouse strains. CD1's an outbred mouse. The 129S, if you're making a genetically engineered mouse model, it's usually done using this strain because they're very permissive for that purpose. And then you'll back cross them onto a black 6 or a Balb C background. These are fully immune competent, of course, and if you're wanting to get onto your efficacy studies, if you're using human cells or a non-rodent cell line, you have to use the immune compromised mice. What used to be the gold standard, has been around for so many years, was the nude mouse, which is deficient in T cells. So it would grow a lot of different kinds of tumors. And it happens to be nude because the reason they're deficient T cells is they lost a gene encoding for thymic development, which also is close to the hair gene. So that's why they're largely hairless. But these mice aren't used as much anymore because there's a lot of extrathymic T cell education that can happen, and these mice get leaky after about 3 months of age. You can start to see functional T cells in the periphery. So the transition has really been to the latest, greatest flavor of immune compromised mouse, the NSG or the NRG mouse. There's lots of different kinds in between these two, but NSG is what has really revolutionized the immune competent investigations. This stands for the non-obese diabetic SCID IL-2 receptor common gamma chain knockout. I think I got that right. And these mice, really the only normal immune cell they have are neutrophils. And so now the unique thing about NSG versus the NRG is that the NSG uses the PRKDC gene knockout, which is a general global DNA repair enzyme that fixes double-strand breaks, which is required for the maturation of T cells and B cells as they generate and change their T cell or B cell surface receptors. Now, the one drawback of the NSG then is, since it's a global DNA repair mechanism in the mouse, if you're doing genotoxic drug studies or using irradiation, these mice are very susceptible to that because none of their cells can repair those double-strand breaks. And what the NRG accomplishes is that in order to eliminate the lymphocyte compartment, they use the RAG1 knockout, which is required for repairing the double-strand DNA breaks, but only in T and B cells. So the NRG mouse itself has a normal radio-resistant phenotype, but in the immune compartment, they don't. And so those mice, if you're doing genotoxic studies or studies using irradiation, those mice are a much better platform to do your studies in. All right, and so the studies that the biological evaluation core does is, of course, the simple toxicity studies. That's the maximum tolerated dose. If you just got one dose, you wanted to test your drug for what is the single highest dose you can get. That's a maximum tolerated dose study. Or you can do the chronic MTD, where you're going to be perhaps dosing like you would over the course of a treatment, maybe daily treatment. And also looking at histology to see if there's any histological evidence of organ damage, and doing serum chemistry. Now, we don't do the histology or the serology ourselves. We simply collect the tissues, and then we'll interface with the histology core or with the clinical pathology lab in the School of Veterinary Medicine for the serology. And also, along with that, becomes the optimization of the dose and the route for your drug. One of the biggest considerations is solubility. That's a big problem, because generally the less soluble it is, the more active it is. But so the challenge for chemistry is keeping these drugs activity and making them soluble. Generally, what we like to administer the drugs in is a solution of no more than 10% DMSO, 0.08% between 20 as a surfactant, PBS, but sometimes that's not possible. And so maybe we'll have to use oral gavage, or we can use a little bit larger volume to solubilize the drug and go intraperitoneal. Or in some drugs really require to be induced or to be injected intravenously. Also, consideration here is the half-life of your drug. If it's got a particularly short half-life, you may want to be injecting it every day, perhaps twice a day. And if it would require even more than that, you probably need to optimize the drug so that it's got a little bit longer half-life. So from this has really been the pharmacokinetic studies, which would be, regarding the drug, the absorption and distribution, metabolism, and excretion once it's been injected. Of course, if you're injecting IV, absorption is 100%, you don't measure that. But if you go any other route, you want to see how quickly it gets into the bloodstream, what organs it goes to, how much stays in the bloodstream, what are the metabolites that are formed, and how is it excreted? Is it through urine or feces or expired air? So pharmacokinetics is really determining what the drug does to the body. And then also there's the pharmacodynamic studies, which is what, so pharmacokinetics is what the body does to the drug. Pharmacodynamics is what the drug does to the body. And this is the the biochemical and physiologic effects through receptor binding and looking at post-receptor effects, other chemical interactions. And then, these are fairly straightforward standard tests by and large, but where we really get involved and spend most of our time is doing the efficacy studies of how well does your drug actually work. And many times we go from toxicity right to efficacy, because the thought would be, well, if the drug doesn't work very well, then really who cares about these other steps, because they can take a lot of time. So we usually just investigate the toxicity, figure out what your dose range would be, and perhaps optimize if you need to, and then get right to efficacy. Although there are arguments for doing efficacy studies last. Of course, if you have a short PK, you'd like to know that first. But those are the studies that we do on a regular basis, and most of the time we are doing efficacy studies. And so the available strains that we
Ben,
use.
can I ask the question?
Absolutely.
Sorry, could you go back? I just put this in chat, but so I have a question about your toxicity, the work you do for toxicity studies.
Okay.
On your previous slide.
Okay, just a second. Why can't I go back? My arrows aren't working.
Okay.
Oh wait.
So
after this one, I think. There it goes.
There we go. So if we wanted to take mice and give them some drugs, different routes, and different dosing, you would do that, and then you would get Do you be able to pull their organs,
Right,
blood collections that we
Yep.
could do for serum chemistry,
correct.
pull those organs? You or someone would do the histology and the histopath?
That's right. Yeah, my core does not do that, but we will interface with the histo core that does.
But I wouldn't have to call them to do it. In other words, if I set up a study with you, I would hand it off to you and then it would go through the process and.
Correct. We could do it either
Okay.
way, and I give you that option. If some people prefer, say, hey, just give me the tissue, I'm going to send it to them when I get time. I don't want to do that right now necessarily.
Okay,
Sometimes they want us to go right to it, and in which case we'll take it there ourselves.
okay. And if you were to do this, do I need, if I have it on my IACUC for here,
Mhm.
do I need approval at Purdue?
Good point. No, you don't, because all these studies are done through our own approved protocol. And as a result of that, we're the only ones that touch the mice and use them once they come to us. And if we need to, we would transfer to our protocol, but usually we are using the mice we have in our own colony.
Got it.
So I'm quite protective of our protocol because obviously I don't let anybody else touch them. So if you came to me, even though your lab might be capable of some of these things, we don't let you handle the mice.
No, I don't. Yeah, I wouldn't want you actually. Because you would not want it done kind of independent. So if
Yeah.
I said to you what I put in the chat, if I wanted to do that, I would just get with you. Do you have a? You may get to this. I'm sorry if I'm jumping in. Do you have a form, or do I just contact you, or?
Right. So good point. So you probably the best way to start off would be to contact me, and then I understand your studies. We can talk about that. But we do have a submission process. It's called iLab.
Oh yeah. I know.
So I like to talk about things first, make
Yep.
sure I understand the project, what your needs are and stuff, and then I'll say, okay, now you got to get into iLab, submit your request there. But what I do is I'll go talk to my staff, already give them a protocol. You'll give as much detail as you can in that online form, and they can refer back to that if they need, but they'll have what I give them. And but the iLab is so we get an account number and we can keep track of things and communicate with you for charges and things like that.
Right. So you answered my question that I'd really contact you first and then go from there. So
yeah.
All right. Sorry.
Okay, so I think I just started on this one. So the strains we use for these initial toxicity studies are usually the three that I pointed out, the Black 6 or the BALB/c or the CD1 strains. And actually there's been a publication or several actually that showed there's really not a whole lot of difference between these strains, between mouse strains. Largely if you're looking for an MTD or you're looking for half-life, the values are almost always within 80% of each other. And so generally you get the same answers, but really what came out of that, the most important consideration is matching the strain you're going to be doing efficacy studies in, especially if you're doing syngeneic studies. If you know you're going to be using a B6 model, well then do your drug testing in B6 mice or your toxicity testing. If you're going to be doing xenogeneic studies, which means you're going to have to be using immune compromised mice, you could do your tox studies in those mice as well, but generally you're going to choose one of these immune competent strains because they're not as expensive. And also generally, especially with us, our immune compromised strains are in high demand, so we don't generally use them for drug testing, toxicity testing. So then the considerations then in the models that your strain and model you're going to choose is going to be dependent on your disease pathway investigation that what part of that pathway you're looking at. Now, as I said before, we really just largely do anti-cancer studies, but we have done some other ones. But if you're looking at asthma, for example, or diabetes, you're going to choose you're going to know the model that you should be choosing based on your project. And I guess I will also say we really don't stock any special strains other than the immune compromised ones and the wild type ones that I mentioned before. The other considerations then, of course, are the etiology of the human disease and the mouse disease as well. You want to try and match the mouse system as well as you can with the humans. For example, if you're studying renal cell carcinoma and you're looking at a VHL translocation model, you don't want to choose Renka, which is a BALB/c model because it doesn't have the VHL mutation. Then you've also got your genetically altered cell lines. Perhaps you don't care about the mouse strain as much, just matching it with the cell lines. Of course, there's mutant transcription factors like P53, or you've got the polybromo, genes involved in chromatin remodeling, or just the gene products of retinoblastoma, P10 knockouts, those sorts of things. Maybe you engineered your cells. You're going to be putting them into into your either the syngeneic strain or a xenogeneic. But then you can get into now the genetically engineered mouse models where the mouse themselves are genetically deficient, they're either a knock-in or a knockout. And of course, then you've got the conditional knockouts, depending, the knockout is useful for if you're investigating a disease that's present at birth, or one that develops after birth, such as in prostate development, things like that. Also, the considerations are very important, is whether you're going to study orthotopic or heterotopic. Orthotopic being studying kind of in the organ of origin. So if you're, again, renal cancer or prostate cancer, you want to be injecting the cells into the kidneys or into the prostate, or just subcutaneous, which would be a heterotopic location. That's not where the tumor would normally be found. And then if you're going to be doing patient-derived xenograft studies, the PDX studies, lots of times that's going to be using human cells, but you can use other species as well. We also have dog PDX, study of bladder cancer. Dogs are quite similar to humans, and lots of times the model after a mouse, or even in lieu of using the mouse, people will use the dog model. So you can choose your PDX based on whatever your needs happen to be there. And then, of course, if you're wanting to do immune studies, there's the whole humanized mouse field now, which can be done in NRG mice, but these mice being initially made at the Jackson Laboratory, they've chosen their NSG strain to do almost all of their further combined knockout, multi-knockout animals. One of the ones that's a very good model to use is the NSG that are a mouse MHC1 and class 1 and class 2 double knockout. And what this prevents is the graft versus host reaction. So if in just an NSG or NRG mouse, if you take human peripheral blood monocytes or lymphocytes and inject them into that mouse, they may last for about 3 weeks or so, those cells. And then you're going to find that they begin to do a chronic rejection of the mouse, and the mice will die. So by getting rid of mouse MHC, you can get a mouse that'll last for several months before there's any graft versus host issues. And then you've got the other strains where they've made them transgenic for a human stem cell factor, human GM-CSF, and IL-3. If you're doing myeloid investigations, myeloid cell, that's macrophages, monocytes, neutrophils, you can use these mice and you get very good development of the human myeloid system if you're injecting in stem cells. They've also got the human IL-15 transgenics if you're studying NK cells or lymphocytes, and lots of times to have an immune system develop from stem cells, they'll inject the NSG mice with CD34 positive cells from the human. Now, each of these models they're very powerful models, but they also have significant limitations, and you need to be aware of those and consider which models you want to choose. And this is one of the things that I'll go through with people that will come to me with a disease or cancer pathway they want to study, and choosing models and trying to figure out what the literature has shown and where it's at and where the holes are that we need to kind of address to be competitive with publication and funding.
Ben, there's a question. Do you also work with rat models?
Yeah, we do. We use rats not nearly as much, but we do toxicity testing in rats. We haven't done tumor studies in rats simply because people haven't come to us with that need yet, but we certainly are capable of doing that. And then a significant strength of our core is our adaptability, and that pretty much sometimes we get some really very innovative and very, very clever projects, sometimes can be quite crazy. And we may not have the skill set in our core at the time, but what we'll do is make sure we go out and we can get training, or we can figure out how to make it work. And so the things we do on a regular basis is, of course, is imaging, and pretty much anything that's available at Purdue, bioluminescence, MRI, CT, ultrasound, We do those studies on a regular basis. We can do any site orthotopic pretty much. One of the things we've really gotten into lately is intracranial implantations. We've been doing a lot of that, but we do intraprostatic, renal, spleen, kidney. Some of the other things we've done is people have come to us wanting to investigate hyperthermic studies with their gold nanorods, which require the use of the high intensity lasers. We've implanted devices. One device, when it was hit with a certain wavelength of light, would emit UV light, which would then create a chemical shift in one of the other components they were injecting, and it was anti-tumoral. We can do photodynamic therapy. We do focal irradiation on mice, just perhaps a tumor growing in the leg, and we'll just irradiate that. Really, if you come to us with any idea, if it's possible to do, we will figure out a way to do it. We undergo training a lot, either through just the School of Vet Med veterinarians that can teach us how to do things, or like currently, actually 2 of our technicians are at a week-long surgery symposium out at the Jackson Laboratory out in Maine right now. So we'll go out to the Jackson Lab and get trained if we need to. And we do that on a regular basis also, just to stay up on current techniques. And so the important considerations here, which I've mentioned before, is really knowing the limitations of your model. And that's of paramount importance, really understanding what your data do mean and what they don't mean, and what they can tell you and what they can't tell you, and not overstating your data. One of the things I point out is that by the time your drug gets to the clinic, 90% of drugs fail in the clinic. And here's the breakdown for the various reasons of that why. And the 40 to 50% having no efficacy is due to a number of things. Obviously, until you get into humans, you don't know exactly what's going to happen. But also a lot of that has to do with poor drug design, poor experimental design before you get to the clinic. And that's one of the things we try to avoid right off the bat in choosing a model and knowing the limitations. And understanding that PK and PD can be affected by age and nutrition, disease state, any other drugs that are being administered. And while mice are a great place to start, when you're not as well funded for things as perhaps as you would be when you see success, right? But lots of times you're going to be trying to attract industry, and they're almost always going to want to see at least rat data before investing, because mice are not as accurate in predicting toxicity for humans, although they're reasonably good enough. Certainly, that's where lots of times you want to start, but the rats are better, much better, and still cost effective. And I don't know if you've all seen it or heard about it or not, but the FDA just made a change now where it used to be law, came about in the 60s, I think, from the thalidomide fiasco years ago, where they had required animal testing before a drug could be approved. Well, that has now been reversed, I believe, last year. Largely was under pressure for the people who were against animal testing in the cosmetic industry. But the FDA has now said there's no longer a requirement for animal testing before getting your drug approved. But still, with a lot of these therapies for non-cancer diseases, and especially cancer, before you're going to get FDA approval, you're still going to have to test these things in an animal model that's non-rodent. Like, for example, I mentioned before, dogs and bladder cancer. And this new law, I don't think it's going to have much effect on us in the near future or even in the distant future, but no one is really sure what the effect of this is going to be. But for now, it's business as usual, in that you really have to perform animal testing. Now, if you want to ask about our costs, this is how it breaks down here. So if you're a Purdue or an IU Simon Cancer Center member, the subsidized rate, heavily subsidized, is $70 per hour. And our cost structures, we don't charge for individual procedures. We simply, if we use supplies, if we use a box of needles on injections, we charge you whatever that box of needles cost. What we charge for is our labor and the mouse. Our mice, each one we make, is $35 until they change our rates in July. But for now, whatever mouse you're using, it's $35 a mouse, and then plus our labor. And we just keep track of how much time we spend on the experiments for weighing mice and injecting and handling and drug administration, tumor implantation, that sort of thing. If you're a Purdue faculty, but you're not a cancer center member, that's $135 an hour, which is still subsidized. Or if you're a PI or industry, we do some work with industry. Other than those classifications there, then the unsubsidized rate is $370 an hour. And so that the way this breaks down for members, if you're going to be doing toxicity studies, this includes the cost of the mouse, all the labor, everything that happens in our core, harvesting the organs at the end of the study. It's going to run roughly 125 to 175 per mouse, depending on what you want. And that's going to be, project dependent. But for a 10 mouse study, you're looking at $1,250 to $1,750 to get that done. That does not include histology charges. And if you're doing mass spec, lots of times those will equal as much. Those two together will be about what our cost is, perhaps a little more. But again, it's highly project dependent. The efficacy studies, if we're doing, this is anti-cancer efficacy, can run from 200 to 300 a mouse. But again, that's really dependent on what the project requires. If we're, if it's just to inject a tumor once, give drug twice a week, maybe, no imaging, you're going to be running closer to $200 a mouse. But if we're injecting the drug once a day or twice a day, imaging twice a week, then there's radiation, a lot more involved work involved there. Then you're going to be pushing 6,000 or maybe a little more for let's say a 20 mouse study. These are just examples to get you an idea of what our costs would translate into the actual studies. But again, what we do is after we've designed the project, and got the experiments protocol set, then we'll send an estimate to you that's much more accurate. And then you could choose to say, no, that's too expensive or not. But if you're doing the subsidized rate and even the non-subsidized rate, it's still quite less expensive for your initial studies than what a CRO would charge. So in summary, then really, the strain choice for initial toxicity testing is not complex. You're generally choosing the same strain you're going to be using, or just a wild type strain. And the complexity really becomes when you're trying to examine efficacy, depending on what your disease is, the organs that are effective, and how well it's matched up with human disease. And also our core is quite adaptable, and we really will do what we can to make sure we can do your project and answer the questions you want to answer. And then I also get very heavily involved in the writing and conceptual development for grants and manuscripts. If you feel you really need that, most people do, even if they know how to use animals, just to make sure that, and I can help guide you through that if you're not comfortable with the animal portion of writing and funding. And with that, that is all I have for telling you about our core and what the technologies are we use, and how you might use that for yourself or be able to use us if you wanted to.
Great, thank you, Ben. There's a couple questions in the chat. The first one is if you wanted to use a PDX, if you want to do a PDX study, do you have cell stocks, or do the users need to provide PDX cells?
Good question. I should have pointed that out. We have some PDX ourselves. We have, for human, we have prostate, we have bladder, we have small cell lung carcinoma. But let's see, we have renal, I believe. So we don't have a large array, but if the tissue is available, we can usually get it and then initiate the PDX model ourselves for you. Or if you have them, you can provide them for us and we will do it as well. So either way works. And we have a limited collection of cell lines. I would probably say that most of the time people do bring their cell line to us. But before we use it, it has to go through testing for mouse pathogens and so forth. But that's not hard to get done.
Ben, you want to go ahead and ask your question?
Oh yeah, I can do that. Hey Ben.
Yes, hi.
I'm Padu. I'm from the Department of Ophthalmology at the Glick Eye Institute at IUPUI.
Yes,
So
yes.
we do a lot of ophthalmology research. We do like drug development, testing it on ocular tissues for looking at either the cornea
Sure.
or the glaucoma studies or retinal degeneration. So do you do PD and toxicology studies on eyes?
Yes, we do. Yes. So we will harvest, we will inject your drugs and harvest whatever tissues we need to harvest. And then now we are not a histology core, nor do we do mass spec. So then we will interface with one of the other cores to get any of the PK or PD that needs to be done.
Okay.
Or any of the serum chemistry, or with the mass spec.
Because why I ask this question is because most of the drugs that people try, they are topically applied, they're applied on the eye. And then you look at the concentration of the drug inside the eye. So basically you need to have I don't know if your team has the skill to collect the fluid that is inside the eye. So there are two different fluids. One is the aqueous humor, which is in the front, or front in the sense, the anterior chamber of the eye. And
Yes.
then the posterior chamber, you have the vitreous humor, which
Yes.
is like a jelly kind of substance. So do you guys have the expertise in that, or that just asking so that we could?
Of course. So actually my first postdoc was in the Department of Ophthalmology with Tom Ferguson at Washington University. And you're shaking your head. You probably know Tom Ferguson's work,
Yeah.
right? That was my first postdoc where I was doing a lot of the immune tolerance studies in the eye and would dissect eyes. And so I used to have that talent. It's been a few years since I've used it. We have not done ophthalmologic studies. Yeah, it's a hard word.
Okay.
But we certainly could. I'm familiar with it and could certainly get the training and then get the equipment we would need to do those studies. When we'd be happy to work with you to do that if you wanted.
Sure. Yeah. I mean, we collect the fluids from the eye. We have a technique to do it. I'll be happy to talk to you if you guys need any expertise. We can always share it with you.
Yeah, that sounds fantastic. Yeah, we're always happy to collaborate that well as well. Also.
Sure.
Yeah.
Thank you.
Thank you.
And then there's a question, what kind of ADME tox assays are you performing?
Okay, so we really just do maximum tolerated dose studies, and we then will harvest the serum or plasma to do the serology, the serum chemistry, and histology as well. We don't actually perform those assays. We will send those to another core, but we do the drug injections and the tissue harvesting and the things that are needed to do the assays that are required. Just basically simple toxicity. Hopefully that answers that question.
Thank you, Ben. That was a great presentation, and thank you so much.
Thank you, Jill. Thank you, everyone, and enjoyed the questions. Thank you.